that the
you want to share the presentation?
right? Yeah, I just clicked on them. I don't know if it's working now or not. Just a second.
Yes.
You can see it.
Yeah.
You can see my screen. The presentation.
No, I don't. But 1 minute ago, I could see it.
Oh,
So
I need to be staying here.
Yeah, maybe you are doing like a duplicate screen.
Right, that's what I'm doing. Yeah.
Yeah, but please do it duplicate.
One.
Yeah, not yeah. You don't do extend, please, because it's gonna conflict the screens.
Let's see that from this screen that I can open up. Yeah, hold on a second. It did work?
Yes, perfect. Can you hold on for a second? I want to check also with Aaron.
Sure.
Yeah, hi Aaron.
Hello.
How are you doing? Fine.
Yeah.
Okay. Can you please test your screen and camera, please?
Yep.
Perfect. Very nice.
All right. You able to see that?
Yeah.
Okay.
Okay, thank you very much. I will come back to you. And now we can we, Liang, do you hear me, please?
Okay, are you talking to me?
Yes, how are you doing?
Good.
Can you please check with us your camera and the presentation?
Yes. Yes, I mean, I'm here.
Yeah, nice. Okay, perfect. So I'll come to back to you after they finish the first two talks. Okay.
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 Okay. Let me close my camera and yeah. Thanks.
Okay. Thank you. And now we can return to Dr. Hikmat.
Sure.
Yeah. I think we have like a couple of minutes until we can broadcast.
I think you can see it, right? Now?
Yeah, I can see it now, but it's not in the full screen.
Now it's full screen?
Yeah. Now it's full screen.
Yeah. Well, I cannot see you. Okay,
No?
that's fine. No, but when I make it like a full screen, it's just on the desktop.
Oh.
That's why, I guess. That's all right. I mean, yeah.
Okay.
Now it's full screen and then everybody can see me. And when you say go ahead, then I'll go ahead.
Yeah.
You can see me though, that right? My camera is on?
No, I don't see the camera.
Really?
Yep. I think you didn't turn it on. Or...
I did turn it on before. Oh, you can share video when you're done screen sharing.
Sorry?
Well, it says that when you're done with the screen sharing that you can do it. You can turn on the camera. Let's say that.
I guess you can share both.
Yeah, you can see me right now.
Yes.
How about like can you see that presentation?
No, not yet.
See that when it's in desktop, that's why, I guess. So what we are gonna do is that we're gonna close
show the presentation.
Yeah.
Okay.
And then I'll just do the presentation. Can you see the presentation now?
No.
Hold on a second. Hold on a second. Can you do that? Can you see it now?
Yes.
Okay. I think without camera that I will do it.
No
Since-
problem. Yeah, we can like start the camera after you've done with the presentation.
Exactly. Yep, that's fine.
Okay. Actually, I'm very pleased to introduce you all. I hope that we all get the full benefit from these lectures.
I hope so.
I think we can start now. You're ready?
Sure.
Yeah. So good morning and good afternoon, good evening, wherever you are. I would like to thank everyone for being here with us today in the third IPGG symposiums. In the beginning, I would like to welcome all the participants, especially who have been with us before, as well as new to the IPGG symposiums. I hope that everybody get benefit from all the worthy presentations which will be given by the valuable, respectable speakers. Before I get started, I would like to express my appreciation to everybody who helped in making this event come true, especially Dr. Ahmed Salam. Now we can start the second section, which is titled Advances in Plant Breeding and Genetics. This session contains 3 different talks. The first one will be given by Dr. Hikmat Budak. Dr. Hikmat Budak received his PhD from Nebraska, University of Nebraska in 2002. Actually, his work is focusing on using different new technologies like NGS and microRNAs and CRISPR-Cas9 in editing plants. Dr. Hikmat worked and led many programs at many valuable universities like Montana University State and University of Worcester at UK, University of Nebraska in USA. Actually, he has a huge number of publications. It's like 170 publication, which with each index around 60. And he has a massive citation number, which like 21,000 different citations. Please, Dr. Hikmat, you can start now. Thank you very much.
I thank you. Can you hear me well?
Yes, yeah, perfect.
Great, excellent. First of all, hello everyone. I'd like to pass along my thanks to organizing committee for inviting me. It's a great pleasure to be here and then just share some of our research with you guys. And then I will just basically what I'm going to talk about today is just the genome editing in wheat, but in basic structure and as well as some of our research we conducted and we're still working on. So I'll start with one of the code that I really love. I got it from my PhD supervisor before. I have drunk from wells I did not dig. I have been warmed by fires I did not build. And the reason I love this is that science is a team effort and actually global. And then whatever I present here, it's just a research team effort. I'm just presenting or representing all my team members. It's a huge, large members that I work with, and these are all their efforts, and I'm just representing them here. Basically, what I'm going to talk to you today is just the introduction part, which is like what the plant breeding techniques. I know that there are a lot of great speakers lined up, and then they already mentioned maybe a number of them. But then I will talk about some editing in wheat and what are the gene delivery method for wheat, and then some CRISPR application that we conducted. And then now we are working on editing some cadmium gene in wheat as well. That's the last part that I'm going to talk to you today. As you all know, that plant breeding techniques, that when we look at it, 4 techniques really used a different period based on the innovations and biotechnological development really. And then we know that the cross breeding based on the naturally occurring mutants, and then it required the backcrossing and transgene free 8-10 years that you will get a new elite cultivar, or just you're going to integrate the genes or traits to commonly grown varieties as well. There are mutation breeding as well. That tool was great as well, and still that it's on an ongoing research effort on mutation breeding. It's just a random mutation that we are putting in and to create the genetic variation. But again, that the backcross is required, and it'll take 8-10 years to just produce a new variety. And then later on that we know that the transgenic breeding and This is easy, but one of the drawbacks or still that they are discussing a lot, that introducing that the new genes from the other organism is an issue because the foreign DNA is integrated. That's what the debate is. And then also, like, it's really subject to a long-term regulations and commercialization part. Now that we know that I would call that the precision breeding is the genome editing techniques, which require less time and highly efficient, and there's no backcross requirement, and then transgene free as well. So this is something that is, I guess, that the new revolution for the plant breeding tools and next generation plant breeding. That's what I call, and I'm sure that there will be a new tools. It is going to be developed soon as well. So these techniques are used to provide a better life for the human. And then we know that based on the World Health Organization data, there's a 11% undernourished of the world's population. And then 22% of the children, younger than 5 years old and stunned. And 9% of the world population is really severely food insecure. And then 1 in 4 people globally that food secure as well. So all we're trying to do is just using or applying all these techniques to provide a better life and then help people as well. When we look at the top 5 factors of the world hunger, there are 5 critical aspects that we can consider: that the poverty, climate change, war and conflict, and then food shortage and poor nutrition. As scientists and plant breeder and geneticist, I believe that we can easily work on that, the climate change and poor nutrition. So this is our goal my goal actually that I'm focusing on. If you were to look at the climate change part, I will give you some data as well. When you look at the water in, on, and above the earth, you will see that this error, that the little amount of the water that we have currently has been used and we are currently using, that's the freshwater lakes and rivers, compared to that whole wild world. And think about it, I love drinking coffee as well in the morning and then sometimes during the nighttime as well when you're writing papers or grants. So do you know that how many gallons of water that we are spending for a cup of coffee? I'm sure that you can answer that, but I will just answer that the 34 gallons of water that we are spending for a cup of coffee. This is just for food and this is just for coffee. And think about it, and then I know that the data as well, that the white t-shirt that we have, and then to have this t-shirt from the beginning to end, we are spending 700 gallons of water as well. So like a food industry, I'll talk about the agriculture as well, that we are using the water, that existing water, that they compare to whatever that the resources we have, we are spending a lot of water as well. So we need to use any tools available, or we need to develop new tools to be able to provide a better life and to help climate change and save our resources as well. So when you look at the genome editing tools so far, there are 3 main genome editing tools based on the factors that when you compare zinc finger nucleases, TALEN, and CRISPR. And CRISPR currently, right now, that high efficiency, it has low cost, and it has rapid construction and easy to deliver. And there's a multiplexing, which I will give you an example that we are focusing on or we're working on now. And then also—
Excuse me, please. I don't see the screen. I don't know if it's my only problem or the else also don't see the screen.
I can see the screen.
You can see it?
Yes,
Okay, sorry. Sorry for interruption.
No problem, that's all right. And an off-target effect as well, it's important, which CRISPR has a low off-target effect compared to zinc finger nucleases and TALEN. It's been out there, ZFN and TALEN has been already out there, but the CRISPR is new and then and started applying all the plants system. So I just wanted to just chart the outline the CRISPR-Cas system. What we are doing is that this is the basic part at the target selection, and then designing guide RNA and construction vector. And then we have delivery methods that in wheat that I will just talk about in details in a bit, and screening the transgenic lines and validate the mutation as well. And based on the guide RNA design and vector construction, a PEM sequence is important part, which we just can look at which nucleosis or nucleotide or protein that we need to use. And the guide RNA target locus is important part region that we need to really understand, because there are lots of off targets that we can just delete and silence or knock out completely different genes as well. So now we are also focusing on some eCRISPR design. We wrote a new scripts, and hopefully that it'll be software soon, that it'll be out there, that computational CRISPR design and tools is available, that everybody can do it. And promoter, as you know, that is important part, and for the expression part of the gene. And then cloning, what are the cloning part that you are going to focus on, guide RNA synthesis, plasmid that you are going to use. So this research is started really in 2013. This is the, like when I was in Turkey, that I focused on a lot about the drought. This is one of the germplasm that we look at it on the left screen, that you will see that the control plants, I mean, we apply this short-term and long-term or prolonged drought stress, that you will see that the root structure. And what we identified is that there are two main genes based on the RNA sequencing, and two candidate genes that was already available. And then we also identified, and we wanted to focus on and edit this gene and see what's going to happen if we can improve the root architecture system in wheat. And we'll also look at the metabolome or metabolomic analysis when I was in Turkey in 2013, that if we can just engineer some of the metabolite to increase the root architecture system in wheat. And this is what we did. And then two genes and candidate genes on the left screen on the top, that it shows that how to use the target gene, basically that until CRISPR system. And then the bottom part on the left, what we did is that we used the drought responsive elements, and two genes that I was talking about. The other one is the ERF3. And we design on the right side that here, I don't know if you can see the arrows that if I show you. Okay, so that on the left part, and you can see that the plasmid, and what we used as a promoter, different promoters, rice U6, maize UB, and wheat U3, and wheat U6 promoters. And then we isolated, and what we identified is the U6 was working perfectly fine in wheat system, because wheat is polyploid. Is that one of the difficult part is that what promoter is we're going to use, and then how the guide RNA is going to work. And then we all put everything into Cas9 vector, and this is the showing that what we did is for these two genes, and then we did the PCR amplification, everything. And then first thing is that the designing primers from the promoter region, and then also like from the right side of vector to amplify, and we did amplify, and it perfectly worked. And after that, this is what is coming next, is that how we need to really just transfer this vector into plants. As we know that there's a biolistic system, hex system, and agro system that are used for the plants and wheat as well. And it's easy and difficult, like both pros and cons, and limitations and strengths in terms of like delivering some of the vectors. What we wanted to do in our research, we wanted to use all three of them. PEG method, it's easy that in 2 weeks that we blend them and then cut it in the 0.5 millimeter like length. And then after that, we cut the strips. And then after that incubation, that we harvest the protoplast. And then you can just see the cell counts, and then you can use it for for producing a new plants. But cells in this region is really fragile. Sometimes you won't get a plant, or you will get really less regeneration as well. But we did that, and then it worked perfectly fine, and we got some plants as well. And these were just happened in Montana State University when I was there. And then this was the GFP validation too. And then all we are doing is that how the transition efficiency is working. We did the GFP, and what we identified is that the GFP expression in 72 hours is that 70%, which was great. We said the PEG is going to work, and these are the first setup for wheat CRISPR system. And then we also use the particle bombardment system for CRISPR. And what we are doing is that we are isolating embryo and then looking at transferring with the ballistic gene gun. And then on the right side, that the red arrow is clearly showing that there is some kind of like editing that the PBS gene, that's what we edited, and using 3 target as well, and using 3 different proteins as well. Not only Cas9, we use the CPF1 and CPF1 LB and AS as well. And based on this, and we identified that the CPF1 is gonna be better for wheat system using the particle bombardment. I'll show you the efficiency in a bit. And we also tried agrosystem, and agrosystem was working perfectly fine. And eventually in agrosystem, as you know, that we are just isolating the embryo, and then callus generation. Right after that, we just transfer, and then based on the selections, either antibody that we used at idomycin that we used at that time, and then we created the plant generation, and we got the transgenic plants. And this was something that is still ongoing research here in Montana BioAg as well. So that is CRISPR system structure construction for in wheat system that I was telling you that we use the Cas9 CPF1 AS and LV CPF1. And we just wanted to compare which one is working perfectly fine or high efficiency and then effectively in wheat system as well. Because we know that the wheat is polyploid. I'll show you that how challenging the work with wheat compared to many plant species, especially diploid. And these are the construction system our construction vector system that we used. And after this, one of the critical part is that when you design the construction, how we can validate if you edit the gene really. And we used the Sanger sequencing, I'll show you in a bit, but we used also T7 endonucleases. On the left side, you can see that if there's any cut between the genome or DNA, that you will see. T7 is just annealing and re-annealing, healing. Then you will see that if there's any differences, that you will see that, okay, that you added the gene or target region that you are focusing or you're interested in. This is one of the example, target 1, target 2, target 3 regions, and the negative control was just the empty vector. And then you can see that the red arrows is clearly showing that what the cuts on the target 1, target 2, and target 3. And Sanger sequencing is one of the critical part as well, that not only T7 that you are validating that you did edit the gene, but also that we did this Sanger sequencing method, and then looking at that, what is the differences, and if there's a mutation between the wild germplasm or wild genes and region, and then also targeted region as well. So we identified a number of mutation by sequencing as well, that it was clear that we edited these 3 as well, 3 targets as well. And this is the target mutation efficiency I was telling, and you guys, we compared the really Cas9, CPF1, CPF2 from LB and AS. I think that now we can also call it like 12, Cas12A. And the name is not a big deal, but in wheat, when we compare all these three using three target genes or regions, what we identified is that CPF1 is the higher mutation efficiency. So whatever that we target and the efficiency in using that CPF1 instead of Cas9, we identified the higher efficiency of mutation, which is that off target is really less, and we really highly focused or edited that region. And the final part, I'll also talk to you about like a multiplexing before I go into cadmium editing genes. It's simple and easy, and I try to just illustrate it with the right side, and you can see that we designed at least like 2 guide RNAs, and we have a shuttle vectors that we can put each one of them, and then at the end combine each of these vectors in one plasmid, and then using the gateway cloning system, and then we can just look at the final destination of guide RNA and Cas9. So what we can do is that if we wanted to edit the 2-3 genes at the same time, and it's a good part in multiplexing in wheat as well. So we did this in using a couple of the genes that we wanted to identify, and then using 3 genes actually that we wanted to edit. And again, U6 was working perfectly fine as a promoter in our experience and then also research. We designed, and these were the vectors that we were using. And then we assembled each one of them, multiple guide RNA expression in one golden gate. And then we put it in the Cas9 and then multiple guide RNA expression cassettes. And then we apply to wheat, and we transfer to wheat. And hopefully that we will identify some of the results in a month or so. This is what we are working at Montana Bioag right now. Like I said, I mean, it's really working with wheat is a big challenge, one of the biggest challenge, because if you were to look at it, the Arabidopsis has like the genome size is 140 megabases, and 10%, about like 10% is repetitive sequences. Brachypodium, my team also got involved with the sequencing of Brachypodium as well, with a number of other research too, but it's just a 355 megabases, and really that the repetitive sequences is really, really less, and the rices as well. And when you look at the maize, 2,300 megabases, and human is in between maize and barley, that is 5,000 megabases in barley. And if you were to look at on the right side, that like bread wheat, especially like hexaploid bread wheat compared to durum, it's 17,000 megabase and 90% repetitive sequences. It is hard to deal with, and then it's 120-fold of Arabidopsis genome and 45-fold of rice and Brachypodium. This is huge genome, and it's not easy to target only that. Well, it's not easy to just have the higher efficiency for off-target regions as well, or genes. Well, with that, I just wanted to tell you a little bit about the prime editing of heavy metal accumulation genes that we are also focusing on here, Montana Bioag. And cadmium is really dangerous for human health, as we all know, and it's just oxidative stress and dangerous for the mitochondria as well. And in plants, it's really dangerous as well. The reason is I would always consider that as comparing zinc and cadmium, because there's an antagonistic effect between zinc and cadmium. If you have high zinc, and that means that you have a lower cadmium. If you have a higher cadmium, that means that you have lower zinc in the grain, which is really bad for the plant health, and then as well as that the commercialization or exporting and importing of some of the durum meats as well, that it's really dangerous. There's a codex value. If you have like 0.2 milligram higher than that, 0.2 milligram higher than that, that means that you cannot export the durum meat into different regions, and they're not going to accept your durum meat as well. When you look at the zinc, why zinc is important, And looking at it, that 10 leading risk factors, World Health Organization again said that zinc deficiency and iron deficiency is the fourth and fifth level in the ranking. You can look at it. Zinc is what really critical. And one of the examples that I'm going to show you is that on the top part is that the high dark red is showing that the high zinc deficiency. And on the bottom, you will see the child mortality. This is the absolute numbers. It's really overlapping with the zinc deficiency. So that's why we were thinking actually that if we lower the cadmium, not only good for the durum wheat, but it's also important for bread wheat so that we can increase the zinc as well. Lowering the cadmium is going to help increase the zinc transportation. And we wanted to understand that how the zinc deficiency in Montana, when you look at it, it's really high zinc deficiency. And the other thing is that in Montana, soil level is pH soil level is between 7 and 8. Even some regions has a higher zinc level. That doesn't mean that it's available to plants. This is the critical part that we also consider or take into account. Some region soil might have a good enough zinc amount, but because of our pH level is low, that means that zinc is not available to plants, and it's highly likely that the plant is going to uptake the zinc. So the solution is not like giving supplementation or food fortification, which is not affordable for many countries or even like here as well, and not sustainable either. So I believe that we just wanted to focus on decreasing the HMA3, and there are a number of them as well, like heavy metal ATPase as well, so that we can just decrease that or knock out some of them and see that zinc level is going to be increased in the grain. And what we identified is the first, we wanted to look at the presence and absence of this gene, what we have in that number of genes, a number of varieties that we tested. Some had that gene and some of them didn't. And some of them has like the different alleles or different alleles is that been present in some varieties. And so we wanted to focus on a couple of them that we wanted to identify or see that if we can, when we added them, complete the knockout to see that the zinc is going to be increased or also cadmium is going to be decreased. This is the map of cadmium genes. This was the beginning. We just wanted to just added knockout of like exon 3, exon 4, and exon 6, but now we added exon 5 as well. And based on the results, the current results, and we believe that we increased the level of zinc, and the cadmium level was really low, although we found that cadmium was really high in some of the varieties. Hopefully that we are editing some final part of the manuscript as well, that we are, before we submitted the grant, that we will publish this paper, and then it'll be available to the whole community. And so that off-target part was really low, and we used the new computational CRISPR protein design and guide RNA design that we developed here as well, that is going to be online available soon as well, that people can design any guide RNAs from wheat, including durum wheat as well. So we believe that the improved transformation efficiency in durum wheat is now getting better and better, that we use the different transformation efficiency, especially like recently Nature Biotechnology, some of the group Johnny Ness Center, and as well as like UC Davis just proposed, and then we use that method as well. And it's going to be great for durum wheat too. With that, I will stop, and I'd like to acknowledge all the team members. First of all, I couldn't list everybody here since this research started in 2013, and we still continue. And there are a number of great team members that work on this research and still continue working on that. And I'd like to acknowledge Montana Wheat and Barley Committee, Montana Grain Growers Association, and when I was in Montana, that Serial Genomics Lab that I set up, And then I also acknowledge USDA for currently providing some of the funding resources too. Again, this is a team research, team effort, and I'd like to uh, really acknowledge all of my team members and thank them as well. With that, thank you so much again, and I would be happy to address any question that you might have.
Thank you very much for this interesting talk. Actually, it was very interesting. I think we have a question here. It's from Dr. Ahmed Salam.
Hello, Dr. Hikmat. Thank you for joining us.
Thank you. I thank you.
So I have a question about the gene editing in wheat. After editing the gene, the target gene, did you test the effect of other yield traits, induced quality, or as the gene has major effects and minor effects? So I am just afraid of editing a gene and affecting other important target genes in such as yield traits or something like that.
That's a great question, really, Dr. Ahmed. Yeah, I think we didn't test all of them, but I believe that for the cadmium genes that we tested almost all quality traits as well, like a mixing dough. It was easy. But I agree with you for some of the traits when you added it, you need to test all the yield and use quality traits. Um, but our research currently that we wanted to set up the protocol for wheat research, editing protocol, now, depending on the genes, we just edited 2 drought genes, and based on that, yield were still same, but still that was just a greenhouse testing, which is not reliable. Okay, we need to take him out to the field to see that if yield is really affected or not. But in use quality, that I would say that for cadmium it was okay, at least for HMA3 and 3 other genes that we tested or we edited. I hope I answered your question. Was that the
Yes, yes,
question?
of course. Yeah, I have another question also.
Please.
Uh, have you used genes that were detected by genomide association study? If I have like candidate genes that were detected by GWAS, uh, are there requirements for choosing those genes like high phenotypic variation explained by SNPs or you know so have you used genes
Okay,
from
yeah. Sorry, go ahead. Yeah, my understanding from your question is that we haven't tested some of the GWAS related genes. And what we are doing is that currently, Dr. Ahmed, we are just doing single RNA sequencing, single cell RNA sequencing. And we're trying to identify the really gene is really have a big effect on the phenotype. If we don't see
yes.
the phenotypic effect, which is something that I would say we're wasting our time. I haven't selected or choose so far any genes from GWAS, um, but in the future, maybe that I will collaborate. But I just wanted to focus on some of the genes that I've been already tested for a long time. Like I said, that these research started like 2013 when I was even in Turkey. So that's why I just wanted to focus on the genes that I've been already working on and seeing the phenotypic effects. Did
Okay,
I
thank
answer your
you.
question? Is that the question?
Yes, 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 yes.
Okay.
Thank you so much. Very interesting.
I thank you.
We have another question here from Hafiz Omar. So he's writing that it's very informative lecture. Do you find any pleiotropic effect on HMA3 gene after knockout?
We haven't tested yet. Well, I believe that there could be, but I think that for editing, I don't believe that this is an issue. That's my opinion for now. Until I just prove it, maybe my opinion is going to change. Anyways, but so far, as soon as that I see that the phenotypic effect or phenotype that is affecting, which is like a lower cadmium or higher zinc, I will go for it. Yeah, and then test in the field. That doesn't mean this is the end of the research, quite frankly. I would say that CRISPR editing system is great system and everybody should use it. But I believe that it has to be a kind of like full package. You need to go into the field and test it, like the first question, test it for yield and yield related traits, and also end use quality traits. Then you will make sure that, okay, this is one line that or genotype that I like to use and send out to farmers for their use. I hope I answered the question.
Genome editing is gonna it's gonna reach, like, are we going
Sorry,
to
I didn't
reach
hear the
different
first
levels?
part. I didn't
So
hear
how,
the first part.
sorry,
Great.
how far we can go further with genome editing? Like, are we going to I don't know. So now we can do gene transfers and gene editing. And do you think that in the future we will have like more more kind of transfer or editing? So I don't know.
I think we will be able to use all of them. I mean, whatever that is required for our research purposes, not only for that, and then eventually that for the final product. I would just say that if you are really focusing on product focused science, then you need to have a system. So in the academia, and I've been working in industry, what I like to say is that maybe this is the differences that I really clearly understood right now, maybe, or I'm trying to understand. And if you want to focus on like one great wheat variety that is going to be good for the region, I would say the targeted region, not for the whole wide world. Everybody has to focus and use whatever the available tools and to have that type of product for farmers. And I just want to emphasize that we need to have a system, which means that starting from editing, selection of the genes, editing the genes, what is the best of the delivery method, agro or particle bombardment, to effectively and efficiently provide the final product, and test them in the field, and eventually give it to farmers, which means that this is a good system that we need to focus on. And for any region, I think plant breeders or scientists, plant scientists and geneticists and breeders that need to focus on and then understand their system for targeted region and what is the best system that they can use and they need to use. And
question
yeah.
here?
Pardon me?
Yeah, it's from, I don't know. Can you unmute your mic and ask the question, please? Allow me out.
I have to allow this. Okay, sorry.
Yes.
Okay, now please you can. Yeah.
Yeah, I'd like to thank you, the organizing committee, and I'd like to thank Hikmat for this very nice and informative talk. I have a little bit short question. Might be I lost some valuable information. I would like to ask about the effect of the genes copy number. As we know, wheat is a hexaploid, and if you edited 1 or 2 copies of a target gene, what about the effect of the remaining copies of the same gene? For example, if you edited the gene for cadmium uptake, if you have edited 2 or 3 copies, what about the remaining 3 or 2 or 4 copies of the 6 copies of a gene in wheat?
That's a great question. That I just wanted to say that that's where the multiplexing comes into play. And thank you for asking this question. So that multiplexing is going to help for polyploid genome like wheat. So we're not just editing that one copy of the gene, and then that's what the multiplexing that we are focusing on, to edit the 6 copies of the genome. So that's the important part, and that's the challenge working with wheat. So, but the multiplexing is going to overcome that issue.
Great, thank you. Thanks.
I thank you.
I think now no more questions. Thank you very much, Dr. Hikmat, for this very interesting talk. Now we can move on to the next talk, which is going to be by Dr. Aaron Carter, professor and the chair of wheat breeding and genetics at the Department of Crop Soil and Soil Sciences at Washington State University.